Assalamu alaikum. Today we want to talk about chapter 4, the three-dimensional structure of proteins. The chapter outline will be as follows. Part 1, we will talk about protein structure and function. In 4.2, primary structure of proteins. In 4.3, secondary structure of proteins. And this concludes part 1. Then in part 2, we will talk about both tertiary and quaternary structure of proteins. Now, protein structure and function. Now, biologically active proteins are polymers consisting of amino acids linked by covalent peptide bonds. Many different conformations, or referred to as three-dimensional structures, are possible for a molecule as large as a protein. Of these structures, one or at most a few have biological activity. These are called native conformations. Many proteins have no obvious regular repeating structure, and as a result, these proteins are described as having large segments of random structure, also referred to as random coil. The term random is really a misnomer because the same non-repeating structure is found in the native conformation of all molecules of a given protein, and this conformation is needed for its proper function. And because proteins are complex, they are defined in terms of 4 levels of protein structures. What are the levels of protein structures? Primary structure is the order in which the amino acids are covalently linked together, or we say it refers to the linear sequence of amino acids in a polypeptide chain, and the chain is read from the N-terminal end to the C-terminal end. The primary structure is the one-dimensional first step in specifying the three-dimensional structure of protein. Now, secondary structure refers to the ordered three-dimensional structure in space of the backbone atoms in the polypeptide chain. We have the alpha helix and beta pleated sheet are two different types of secondary structure, which we will talk about as we go. Now, in many proteins, the folding of parts of the chain can occur independently of the folding of other parts. Such independently folded portions of proteins are referred to as domains or super-secondary structures. Now, tertiary structure refers to the three-dimensional arrangement of all atoms in a protein, including those in side chain and in any prosthetic group. However, in the quaternary structure, a protein can consist of multiple polypeptide chains called subunits, and the arrangement of these subunits with respect to each other is referred to as the quaternary structure. Now, the interaction between subunits is carried out by non-covalent interaction, such as hydrogen bonds. We studied earlier, chapter 2, electrostatic attractions and hydrophobic interactions. Now, for 2, the primary structure of protein. Amino acid sequence helps determine the three-dimensional conformation of a protein, which determines its property. Determination of this sequence is a routine operation in a classical lab by a machine called amino acid analyzer. Now, changes in one amino acid residue in a sequence can change the biological function. Example is found in the hemoglobin associated with sickle cell anemia. In this genetic disease, red blood cells cannot bind oxygen efficiently. The red blood cells also assume a characteristic sickle shape, giving the disease its name. Then these sickle cells tend to become trapped in small blood vessels, cutting off circulation and thereby causing damage to the organ. All these results from a single amino acid substitution in the beta chain of hemoglobin by substituting glutamic acid residue number 6 by valine at the same position. So when you replace charged amino acid with non-polar, then aggregates will occur in red blood cells. Red blood cells become sickle. So again, the primary structure of a protein determines the other levels of structure. And here we saw an example of a single amino acid substitution that gives rise to a malfunctioning protein, such as in the case of sickle cell anemia. Now in section 4.3, secondary structure of proteins, now refers to the hydrogen bonded arrangement of the backbone of a protein or the polypeptide chain. The nature peptide chains are linked at opposite corners by swivels, and each amino acid residue has two bonds. Within each amino acid residue are two bonds with reasonable free rotation. Now the bond between the alpha carbon and the amino nitrogen, as you can see here, they are referred to as phi and psi. These are the two, and the bond between the alpha carbon and the carboxyl carbon of that residue. Then here we have free rotation, free rotation. These are the two angles. Now there are two kinds of secondary structure that occur frequently in proteins. These are the repeating alpha helix and the beta pleated sheet or beta sheet. Now the phi and psi angles repeat themselves in contiguous amino acids in irregular secondary structure. The alpha helix and beta sheet are not the only possible secondary structure, but they are by far the most important and deserved a closer look in secondary structure. Now, as I said, we have two examples on secondary protein structure. The alpha helix, stabilized by hydrogen bonds parallel to the helix axis within the backbone of a single polypeptide chain. The helical conformation allows for a linear arrangement of the atoms involved in hydrogen bond, which provides maximum bond strength and makes the helical conformation very stable. Now, features. What are the features of the alpha helix? Coil of the helix is clockwise or right-handed, and also there are 3.6 residue for each turn of the alpha helix, and the pitch of the helix, which means the linear distance between corresponding points on successive turns, is 5.4 angstrom, or simply 0.54 nanometer in the SI unit. So here, if we look, this is the alpha helix, four different graphic representation of the alpha helix. Here, this is the turn, so we have 3.6 amino acid residue per this turn, and the pitch refers to the linear distance between two turns, like this. This is equal to 5.4 angstrom. Notice how small is this distance. Now, generally speaking, proteins have varying amount of alpha helical structure, varying from a few percent to nearly 100%. Several factors can disrupt. Then again, alpha helix, each peptide bond is trans and planar. CO of each peptide bond is hydrogen bonded to the NH of the fourth amino acid residue. The alpha helix is stabilized by hydrogen bond parallel to the helical axis within the backbone of the single chain. All the side chains lie outside the alpha helix. Now, several factors can disrupt the alpha helix. Now, the amino acid called proline creates a bend in the backbone because of its cyclic structure. It cannot fit into the alpha helix. So, rotation around the bond between the nitrogen and the alpha carbon is severely restricted, and 2 proline's alpha amino group cannot participate in intra-chain hydrogen bonding. Other localized factors involving the side chains include electrostatic repulsion, strong electrostatic repulsion between close amino acids with similar charges, lysine positive, arginine positive, glutamate negative, aspartate negative, and also by steric repulsion of bulky groups. This is called crowding, caused by the proximity of bulky side chains. When these amino acids, any two of these amino acids are in a close proximity to each other, they lead to steric repulsion. This is also called crowding. Now, in alpha helical conformation, all the side chains lie outside the helix, so there isn't enough room for them in the interior. The alpha carbon is just outside the helix, and crowding can occur if it is bonded to 2 atoms other than hydrogen, as in the case with valine, isoleucine, and threonine. Now, what about the second example on secondary structure, the so-called the beta sheet? Now, hydrogen bonds between peptide chains can be inter or intra-chain, and this gives rise to the zigzag structure and are perpendicular to direction of the protein chain. These polypeptide chains lie adjacent to one another. Now, if chains run in the same direction, the sheet will be parallel. If they are in opposite direction, then it's anti-parallel. Again, when alternating chains run in opposite directions, an anti-parallel pleated sheet is formed. Now, the hydrogen bonding between peptide chains in the beta sheet gives rise to a repeated zigzag structure, as I said. Here, the name pleated sheet. The R groups alternate above and below the plane. Each peptide bond is S-trans and planar. So here, in this figure, you see the 2 examples on beta pleated sheet, the parallel here, and this is the anti-parallel. Notice that the hydrogen bond occur between 2 chains, and they run perpendicular to the direction of the chain, and this is different from the hydrogen bond in the alpha helix, which runs parallel to the alpha helix axis. Now, this figure shows you the 3-dimensional form of the anti-parallel beta pleated sheet. Note that the chains do not fall back on each other, but are in a fully extended conformation. What about irregularities in regular structure? Now, other helical structures are found in proteins. These are often found in shorter stretches than the alpha helix, and they sometimes break up the regular nature of the alpha helix. The most common is the 3-10 helix, which has 3 residues per turn and 10 atoms in the ring formed by making the hydrogen bond. So we have irregularity in alpha helix and irregularity in beta pleated sheet. So in alpha helix, we have, the example is called the 3-10 helix. We know that there are 3.6 amino acid residue per turn, but when it is 3 amino acid residue per turn, this is irregularity code. This is in alpha helix. What about irregularity in beta sheet? This is called a bulge. A bulge is a common non-repetitive irregularity found in anti-parallel beta sheet, where it occurs between 2 normal beta structure hydrogen bonds. and involve two residues on one strand and one on the other, as you can see here. Don't worry about the various classes of beta bulge. Just know what is the definition of beta bulge, and it is an irregularity in beta sheet. Common, non-repetitive, irregular secondary motif in antiparallel beta sheet occurs between two normal beta hydrogen bonds and involve two residues in one strand, etc. Now, irregularity, two types of irregularity, one in alpha helix called 310 helix, and one example in beta sheet called beta bulge. Now, protein folding requires that the peptide backbones and secondary structure be able to change direction. Often a reverse turn marks a transition between one secondary structure and another. So, this is called reverse turn. Parts of proteins where the polypeptide chains fold back on itself. Because of for steric reasons, glycine is frequently encountered in reverse turn, because glycine is a small, in which the polypeptide chain changes direction. Now, the single hydrogen of the side chain prevents crowding. But proline is also another possibility to be found in reverse turn because of its cyclic structure. So if we look at this figure here, these are the structure of reverse turn. Now this is called a loop. This loop, مثلا, we take a loop of protein consists of 1, 2, 3, 4 amino acid residue. Now here look residue number 3. Now the side chain, this is the golden ball represents the R group. Now R group is outside the loop. So because of that, any amino acid can have position number 3 in type 1 reverse turn. However, in type 2 reverse turn, what happens here? The loop will turn by 180 degrees. So now the R group is inside. Now size matters here, and the only amino acid can fit into position 3 is glycine. So this is called type 2. Then in type 2, position 3 is occupied by glycine. However, in type 2 proline containing, as we said, the proline is a candidate for reverse turn as long as the side chain lies outside the loop, so it occupies position number 2. So these are the 3 types of reverse turns. Now we come to the so-called super-secondary structure and domains. Super-secondary structure results from the combination of alpha and beta strands, and we have 4 types of super-secondary structure. Beta, alpha, beta unit. 2 parallel strands of beta sheet are connected by a stretch of alpha helix. The alpha-alpha unit contains 2 antiparallel alpha helices, also called helix, turn helix. Beta meander antiparallel sheet is formed by a series of tight reverse turns connecting stretches of the polypeptide chain. And finally, Greek key formed when a polypeptide chain doubles back on itself. And these are shown here. This is type 1 called beta in green, alpha, and another beta. Notice that the 2 beta run in the same direction. They are parallel and they are connected by a stretch of alpha helix. And this is the symbol. Now, this is the most common super-secondary structure. Now, when you have alpha-alpha, notice that alpha-alpha, the 2 are antiparallel, means they are run in opposite direction. So this alpha-alpha, this type is the most energetically favorable one. Then you have the beta minder, and the Greek key. Now we want to define a motif. A motif is a repetitive super-secondary structure. Some of the common smaller motifs are shown inside the book in figure 4.9. These smaller motifs can often be repeated and organized into larger motifs. Now, protein sequences that allow for a beta minder or a Greek key can often be found arranged into a beta barrel in the tertiary structure of protein. Now, these motifs provide information about the folding proteins, but they do not help predict the biological function of the protein. Now, proteins with same type of function have similar protein sequences consequently. Now, domains with similar conformations are associated with the particular function. So this figure shows you some beta barrel arrangements. Don't worry about this figure, just look at it, don't memorize it. Now we come to the so-called the collagen triple helix. Now, collagen is a component of bone and connective tissue, is the most abundant protein in invertebrates. It is organized in water-insoluble fibers of a great strength. A collagen fiber usually consists of 3 polypeptide chains wrapped around each other in a rope-like twist or triple helix. This triple helical molecule is called topocollagen with this molecular weight. Each of the 3 chains have a repeating sequence of the 3 amino acid residue, X-proline glycine or X-hydroxyproline glycine. Now, proline and hydroxyproline can constitute up to 30% of the residues in collagen. Hydroxyproline is formed from proline by a specific hydroxylating enzyme after the amino acids are linked together. Hydroxylysine also occurs in collagen. First position X can be occupied by any amino acid. Now, in the amino acid sequence, every third position must be occupied by glycine. So, glycine accounts to about 33% of collagen residue. And now, this is a triple helix. This figure shows us a triple helix. You see here, all 3 individual collagen chains are helical that differ from the alpha helix. The 3 strands, these, are held together by hydrogen bonds involving hydroxyproline and hydroxylysine. Now, they found that with age, collagen helices becomes cross-linked by covalent bonds formed by reactions of lysine and histidine residue. Unhydroxylated collagen is less stable than normal collagen. Scurvy results from fragile collagen. Now, scurvy symptoms such as bleeding gums and skin discoloration are the result of this fragile collagen. The enzyme that hydroxylates proline and maintains the normal state of collagen requires vitamin C or ascorbic acid to remain active. So this scurvy is caused by dietary deficiency of vitamin C. Now, what are the 2 types of protein conformation? We have fibrous and globular. We start with fibrous. Contain polypeptide chains organized approximately parallel along a single axis, consist of long fibers or large sheets. They tend to be mechanically strong. They are insoluble in water and have the ability to dilute solution, play important structural roles in nature. So they have this only function. And examples are keratin in hair and wool, collagen in connective tissue, including cartilage, bones, teeth, skin, and blood vessels. The other type is called globular proteins, proteins in which the backbone folds on itself to produce a more or less spherical shape. Most of their polar side chains are in the outside and interact with the aqueous environment by hydrogen bonding and ion dipole interactions. Most of their non-polar side chains are buried inside, have substantial sections of alpha helix and beta sheet. They tend to be soluble in water and salt solutions. They have compact structure. In addition, they have various functions. A globular protein could be can transport oxygen as in hemoglobin, or it can be an enzyme which catalyzes a reaction, can be a hormone, etc. So globular proteins have various functions as opposite to fibrous proteins which have only one structural function. So this figure shows us the comparison of the shapes of fibrous and globular proteins.